Hello everyone, welcome back to my video channel. In this video, I'm going to solve the KSET Life Science question paper from 2013 to 2021. Okay, and topic is methods in biology. Okay, and on this topic, that is methods in biology, I have already made two videos as a part 1 and part 2 video. Okay, and this is the part 3 video. Okay, which is going to continue with the 27th question. Okay, and if you guys watched that video, that is well and good. If you didn't watch that video, you can refer those videos, because in those videos also I have completed the previous questions from 2013 to 2021 of the topic methods in biology. Okay, now without making any delay, let's start with the 27th question. Okay, and the question is like this. Dash is a method of genetic analysis that can be used to estimate evolutionary rates and time scales using data from DNA or protein. Okay, option A RAPD, option B SSLP, option C SNP, option D molecular clock. Okay, and as of I know, these or techniques, okay, used in many experiments. Okay, these are the techniques to find out informations. Okay, so now let us know what are all these. Okay, here the first one here is the first option is option A, that is RAPD. Okay, what is this RAPD? RAPD is the long form of RAPD is randomly amplified polymorphic DNA. Okay, it is a randomly amplified polymorphic DNA. Okay, it is this one, and this is pronounced as rapid. Okay, and this one is used to identify genetic variations. Okay, this one is used to identify the genetic variations. This is if you have to say, this is nothing but a type of PCR reaction. Okay, that is polymerase chain reaction. Okay, okay, so that's why this one is not. This one RAPD, this is used to identify the genetic variation. Okay, but here particularly they are saying the question is about estimate the evolutionary rate they are asking. Okay, but these are not gives the evolutionary rate. Okay, that's why that is not the correct answer. The next one SSLP. SSLP is if you have to say long form for this is simple sequence length polymorphism. Okay. These are nothing but these are array of repeated sequence that display length variations. Okay, and if you have to say these acts as DNA markers. Okay, these are short randomly repeated DNA sequences that are present in variable copy numbers at a given locus, and these usually scattered throughout the genome. Okay, and you can see there are two types of SSLPs. That is the first one here is the mini satellites. Okay, those are nothing but VNTRs, that is variable number tandem repeats. Okay, the another one is the microsatellites, that is STRs, that is simple tandem repeats. And these also SSLPs are also they are also used as genetic markers with PCRs, that is polymerase chain reaction. Okay, so these are merely acts as the genetic like DNA markers or genetic markers. Okay, but these never gives the what is that evolutionary rates or the differences between the DNA of the species. Okay, so and the next one is SNP. SNP is single nucleotide polymorphism. Here in this case, what happens? This SNP, a DNA sequence, it is a DNA sequence variation that occurs when a single nucleotide like adenine, thymine, cytosine, or you can say guanine, right? These are nucleotides, right? These nucleotides in the genome sequence, if these get altered, okay, and the particular alteration is present at least 1% of the population, okay, those are called as SNPs. Okay, there is only see here it is single nucleotide polymorphism, to make you to understand very easily. So as in the name indicates, you can say single nucleotide polymorphism. Single means one. Okay, there is only one change in the genome sequence. Okay, that is SNPs, and these SNPs these are going to used to identify the diseases causing GNS in humans. Okay, so by this small explanation, I hope you got some idea that these three are not the correct answers. Okay, because we left with the last one, that is D. So obviously the last one, molecular clock, will be the correct answer. Now let us know what is this molecular clock. Okay, the molecular clock, we are going to study this in evolution topic. Okay, so now let us know what is this molecular clock here. See, molecular clock means we know that all life, means all living things, inherit the information in the form of genetic molecules, right? Usually in the form of DNA, okay, from the previous generations. And this DNA is, that is going to access genetic material. Gradually it changes over the time. We know this, right? And that is the key point here. This can be used to measure how long ago evolution events took place. Okay, that is the thing, the key point they used to identify the evolutionary rates. Okay, that concept is called as molecular clock. Now let us, I will explain a little bit more here. Here, molecular clock, this concept is based on the heredity and mutations. Okay, mutations, especially neutral mutations. Okay, and this molecular clock, it has another two names. It is also called as gene clock, or it is also called as evolutionary clock. Okay, and it is proposed by Emil Zuckerkandl and Linus Pauling. Okay, these two scientists proposed this molecular clock or evolutionary clock. Okay, the concept of this molecular clock is based on hypothesis that the DNA and protein sequences, okay, the DNA and protein sequences evolved at the rate that is relatively constant over time and among different organisms. Okay, and this constancy is used to estimate the length of the time that various organisms have been diverging from one another by measuring the degree of differences between the two sequences. Okay, if you have to make it understand, for example, if you have to make it understand, if you have to take this one, if you have to say we have if the species obviously have a common ancestors. Okay, common ancestor. Take this is the species 1 or species A, and this is species 2. Okay, and these two species, okay, will diverge, they will different, okay, but not suddenly. It takes millions of years, okay, like more than 25 million years, okay, to differentiate by themselves. Okay, that much time it will take. Okay, so estimate this one. Okay, that concept is called as molecular clock. Okay, and for example, like if you take species A, okay, which is going to differentiate from B. Okay, how it is possible? But how you can going to find out species A is different from species B? By the basic difference in the DNA or protein. So here scientists, what they will do, they will estimate the how much time of A and B, okay, will took to diverge. Okay, so that can be calculated by using the difference in the DNA or in the proteins. Okay, by two things, that is by protein sequence or by DNA sequence. Okay, that's why the definition of the molecular clock you can take in the question itself. That is the definition of the molecular clock. Okay, so the correct answer here is the option D. Now let us move on to the next one. Which techniques are used to study the transcription process in the cell? Which is asked in the year 2018. Option A. Here the option A is Northern and Western blot. Option B, PCR and restriction digestion. Option C, Northern blot and in situ hybridization. Option D, Southern blot and ELISA. Okay, so here they are asking about the transcription process. Okay, transcription process. Here they are asking about the transcription process. We know that this transcription, if you have to look at the central dogma here, the central dogma here is the DNA. Okay, DNA forms the mRNA by the process known as transcription, right? By the process known as transcription, and then again here the mRNA is converted into proteins by process known as translation. This is the central dogma, right? Okay, here the question is, which technique are used to study the transcription process? Okay, transcription process, this is mRNA, right? In the cell they are asking. Okay, so in the previous video only I have given some key point how you have to remember the blottings, okay, the blot analysis in the name itself. Okay, a small tip for you to remember for a longer time. Which one I have given? Yes, Southern blot means DNA analysis among the mixture of the many heterogeneous thing. If you have to identify the particular DNA, that is can be done by using that is Southern blot. And then in Northern blot, which is for RNA, and Western blot, which is for protein, and then Eastern blot, which is for post-translation modification. Right? So these are the 4 key points I have given here. And here the question is they are asking the study the transcription process in the cells, right? So option A, northern and western blot. Option B, PCR and restriction digestion. Option C, northern blot and in situ hybridization. Option D, southern blot and ELISA. So four which I have explained just now. So by that we got to know that option C will be the correct answer, okay? Because northern blot is the one which gives the particular RNA, right? Okay, so that's why these will obviously, if you got to know just a little bit idea, so you can eliminate the rest of the things, right? So this will be the correct answer. And ELISA will not be the correct answer, which is because the enzyme linked immunosorbent assay it is. So it is not about that. And PCR, it is polymerase chain reaction. It is just a amplification or multiplication of the DNA sequence. Okay. And if you have to say about in situ hybridization, this is to find out, to determine the presence and absence of DNA and RNA sequences of the interest. That's why. So if we have to find out the DNA RNA sequence, so this one will help. And northern blot, it is for the RNA. So here the transcription process is here DNA is converted into mRNA, right? In case of transcription process. That's why this one will be the correct answer. Now let us move on to the next question here. The flow cytometry is generally used dash, which is asked in the year 2018. First option A is to study localization of protein in the cell, to study the biomolecular interaction, for identification of cell organelles. Option D, for identification of cell types. So in the previous video only I have explained what is this flow cytometry means, okay? But still one more time I will just give a little bit hint here. So this is the picture of the flow cytometry. So if you these are the different types of cells, okay? And this is the detector, okay? By detector we are going to find out the detect and identify, and we can also count the specific cells, okay? By this flow cytometry we can find the cell size, cell count, okay, and cell cycle of the cells, okay? And here, so the correct answer for this is for identification for the in these four options, the correct answer for this is option D, that is for identification of cell types. Now let us move on to the next question, 30th question, which is asked in the year 2018. The question is like this: three-dimensional structure of living cells and tissues can be viewed in dash microscope. They are asking. The option is option A is phase contrast microscope. Option B, fluorescence microscope. Option D, differential interference contrast microscope. Option D, stereo microscope. The first option, if you look at the first option, phase contrast microscope. Here, this one, as the name indicates contrast. This one is used to enhance the contrast of image of transparent and colorless specimens. Okay, so this one doesn't use any three-dimensional structure of the living cells and tissues. That's why this is not the correct answer. Next one is fluorescence microscope. Fluorescence microscope. Fluorescence means you can see the coloring. Okay, again it is. The fluorescent dyes can be used for the antibodies to identify the specimen, right? So this one also doesn't give the three-dimensional structure. So that's why this is also not a correct answer. The next option is that is differential interference contrast microscope. Again, as the name indicates, again I explained while explaining this phase contrast. You can think this here is the contrast. So this is also used to this is the one actually gives the 3D effect only. Okay, that means the pseudo 3D effect. Okay, for the transparent and colorless specimens, but not exactly the three-dimensional structure. Okay, so that's why this is also not a correct answer. The left with the only D option. So D option will be the correct answer. That is a stereo microscope. This stereo microscope which gives the three-dimensional species of the living cells and tissues of the specimen. Okay, so that's why this is the correct answer. Now let us move on to the next question. Here the next question is. Which of the following methods is used to detect the distribution of distribution of the specific mRNAs within a cell? So here which is asked in the year 2018. Option A is RNAase protection assays. Option B, in situ hybridization. Option C, Nodron blotting analysis. D, site mapping. Here option A. That is RNAase protection assays. This one is also called as nuclease protection assay. Okay, and obviously this is a laboratory technique used in biochemistry and genetics to identify the individual RNA molecules in a heterogeneous RNA sample, which is extracted from the cells. Okay, and this one also used to identify and to detect and measure the abundance of specific mRNAs. Okay. In a sample of the total cellular RNA. Okay, that is RNAase protection assay, or you can say nuclease protection assay. Okay, so and option B, that is in situ hybridization. Option C, northern blot analysis. Option D, site mapping. Coming to option B, that is in situ hybridization. As I explained before, in situ hybridization is nothing but to determine the presence and absence of DNA or RNA. Okay, which is it is also used as well as to localize this sequence to specific cells or a chemical sites. Okay, so as I mentioned here, to localize the sequence to specific cells and chromosomal sites. So they are asking, obviously they are also asking the distribution of the specific mRNA within the cell. Okay, this technique, that is in situ hybridization technique, is the correct answer. That is option B. And as we all know, northern blotting, this is used to identify the RNAase. Okay, in a mixture of RNAase. Okay, in a mixture of a sample. Okay, next the last option, site mapping. Site mapping, this is not, we have two types of mapping, that is gene mapping and physical mapping, but I have no idea about this site mapping. I guess this term is not related to this topic. Okay, so option B is the correct answer. Now let us move on to the next question, that is 32nd. The question is like this here, that is goodness of fit is carried out by using option A, chi-square test. Option B, student t-test. Option C, ANOVA test, that is ANOVA, and option D, PCA test. This is asked in the year 2018. So before that, let us know what is this goodness of fit. This goodness of fit we are going to study in statistics. Okay, this goodness of fit test is used to test the if the sample data fits a distribution from a certain population. Okay, for that reason they are going to use this test, and this one which one it is going to use means the correct answer for this is option A, that is chi-square test. This is the one which help in the distribution from a certain population. Okay, and coming to students t-test and ANOVA test, these two are the similar ones. Okay, this one is used to find the population mean between the two groups. Okay, between the two groups, and ANOVA it is more than two groups, more than three groups, like that. Okay, this ANOVA means analysis of variances. Okay, and then option D, PCA. PCA means the long form this is principal component analysis, which is not related to that. So in this, if you remember, goodness of fit test is carried out by using the which test means obviously there is only one, that is chi-square test. Okay, this is the correct answer. Move on to the next question, that is 33. Which of the following techniques could be used to demonstrate protein binding to specific DNA sequence? They are asking, which is asked in the year 2021. Option A, western blot hybridization. Option B, northern blot hybridization. Option C, southern blot hybridization. Option D, electroporetic mobility shift assay. EMSA. Okay, electroporetic mobility shift assay. So here question is, let us look at the question here. Used to demonstrate protein binding to specific DNA sequences, nothing but nucleotide sequence, you can say, right? Okay, the protein binding to specific DNA sequence, that means protein, you can say nucleotides, right? Specific nucleotides. Okay, protein and nucleotides, right? So here we know these three options. So like if you 2-3 minutes back I've explained this, what is western blot, northern blot, southern blot here. So this southern blot to identify the specific DNA, right? Okay, and this northern for specific RNA, and western blot it is for protein. But here obviously these two are not the correct answer, right? So western blot, when coming to western blot here, the protein binding, they are asking not about only identification of the protein, here protein binding to specific DNA sequence they are saying. So that's why this is not the correct answer. So the left one will be the option D, that is electroporetic mobility shift assay. This one will be the correct answer. Okay, so before that let me explain what is this electroporetic mobility shift assay. Electroporetic mobility shift assay is used for studying the DNA binding property of a protein. Okay, and it is based on the observation that the electroporetic mobility of protein and nucleic acid complex is typically less than that of the free nucleic acids. So the correct answer here is the option D. Okay, now let us move on to the next question, 34th question. The question is like this: diagnostic tool that is used to measure and record the electrical activity of the heart is option A, MRI, option B, electrocardiogram, option C, CAT scan, then option D is myelomogram. Here for the first Option MRI is nothing but a magnetic resonance imaging, right? So this one is used to identify the organs and structures inside our body, okay, like brain, spinal cord, like that. This is not nothing related to heart, so they are asking the heart. And the next one here, I will come to this point later, coming to C option here. Okay, I will come to this option later. Coming to CT scan. CT scan is it gives the X-ray images. Okay, it gives the X-ray images of the internal organs which are injured. Okay, so and myelogram. This myelogram is nothing but this is also called as myelography. This actually uses the combination of the contrast dye and X-ray and CT scan. Okay, using those three, combination of those three actually gives the information about the spinal cord. Okay, this is about, so these three are not the correct answers. So option B will be the correct answer, that is electrocardiogram. Okay, which is asked in the year 2021. Next question is 35th question. Here the question is like this: the X-ray diffraction studies conducted by dash were the key to discovery of the structure of the DNA. Option A, McClintock. Option B, Franklin. Option C, Mendelssohn and Stahl. Option D, Cheragov. The correct answer for this is option C. Correct answer for this is option not option C, option B is the correct answer, that is Franklin. The X-ray diffraction studies conducted by Franklin were the key to discover the structure of the DNA. Okay. Now let us move on to the next question, that is 36th question. In oil immersion microscope observation, refractive index of the oil must be the same as that of dash. So if you go through this oil immersion microscope, if you study that topic, so where you can find option A will be the correct answer, that is objective lens. Here the refractive index of the oil should be same as that of the objective lens. Okay. Now let us move on to the next question, that is the 37th question. Choose the correct combination of the molecular marker, markers used in PCR based DNA amplification. Okay, here they are asking these two are also asked in the year 2021. Okay, this is also asked in 2021, and this one is asked in the year 2014. Okay, so they are asking molecular markers, I mean nothing but the genetic markers which is used in PCR based DNA amplification. Okay, the option are like this: RFLP, AFLP and SSR. Option B, AFLP, SSR and RAPD. Option C, RFLP, RAPD and SSR. Option D, RAPD, RFLP and SSR. Here in all of this, option A, let us look at this option A. Option what it says, RFLP, AFLP and SSR. Let me explain this. These things, I will give a small idea for this. RFLP, what is this RFLP? RFLP is nothing but, if you look at the long form here, it is nothing but the restriction fragment length polymorphism. Okay, and it is used to analysis and mapping of the larger genome, and that one is digested with the restriction enzymes. Okay, here in case of restriction fragment length, restriction means restriction enzymes. By using the restriction enzymes, they are going to digest the genome, okay, of interest. Okay, and in case of AFLP, if you come to long form of the AFLP, your AFLP means amplified fragment length polymorphism, and okay, this is not the PCR based technique. This is PCR based technique. Okay, this is PCR based technique that uses the selective amplification of the subset of the digested DNA fragments to generate and compare the unique fingerprints for the genome of interest. Okay, and these SS are nothing but the simple sequence repeats. Okay, these are also acts as a genetic markers which are based on the PCR. Okay, here the one which is not based on PCR based DNA amplification is RFLP. So these two are same. You can, if you look at this here, fragment length polymorphism, right? R and A is different. Here R means restriction enzymes, A means amplification. In this case, what happens? Only the restriction digestion of the genome is going to take place, only one process. But here both restriction digestion of the genome is going to take place, plus there is a PCR based technique is going to add here, both digestion with restriction enzymes and selective amplification of the, okay, using the PCR amplification. Okay, in case of AFLP. Okay, this is different. This is not going to, only the restriction enzyme digestion only going to take place. Okay, and here they are asking as the PCR based DNA amplification. That's why this is not the correct one. So if you look at RFLP in all these three, so here it will be present. So this one, these three will be the correct answer. The left one will be the B answer, that is AFLP, SSRP and RAPD. So AFLP, I just now explained. SSRP means these are also genetic markers or molecular markers that is used uses the PCR based DNA amplification. Okay, and I have explained this in this video only in 2-3 minutes back, that is RAPD. What is this RAPD? Randomly amplified polymorphic DNA to identify the genetic variation, and this is also is a type of PCR based technique. Okay, this is the correct answer. Now let us move on to the next question here. Which of the following forms of DNA travel faster when run together in an agarose gel electrophoresis, which is asked in the year 2014. So I think if this is the agarose gel electrophoresis. Okay, so I'm just drawing it here. So 4 option what they are given is nicked supercoiled DNA, linear DNA, and plasmid DNA. Plasmid DNA is nothing but the extra chromosomal DNA, right? And linear DNA. So linear DNA, you know, this is the linear DNA, right? And here supercoiled DNA will be like this. Supercoiled, it is supercoiled. And nicked means there will be a nick like this. There will be a nicks. Okay, there will be nicks in the DNA, and which is also super coiled. So when all these will be displayed on the electrophoresis, so how the movement they are asking, how much fast they will travel, they are asking. So here the travel of these three, when you look at this, the fastest one will be the super coiled DNA. Okay, the 38th one will be the correct answer. The super coiled DNA will be the correct answer. Okay, so these are the some of the questions which have been asked from 2013 to 2021. I collected almost all questions from the topic methods in biology. So this is the part 3 video, and I have another 2 video that is part 1 and part 2. You can watch all these 3 videos for this topic and prepare well for the examination. And I hope this video is helpful. I just share the knowledge that I have. So I am thinking this video is helpful for you all guys, and thank you for your support. And if you like this video, if you learn something new, please do share and subscribe. Thank you everyone.